Concept 14.3. I figured I'd make the last one short because it has some math in it, so you might want to clear your palate and take a break before you get to the rest of the chapter here. I think there's about 2 more concepts to go here, so again, this chapter is dense, but I don't think the information is too difficult to digest. Concept 14.3 says inheritance patterns are often more complex and predicted by simple Mendelian genetics. So if everything in genetics worked the way that Gregor Mendel saw in his pea plants, things would be far, far easier. But of course, biology can be and tends to be very complex. So genetics doesn't always follow the simple patterns that Gregor Mendel saw in his pea plants. As a matter of fact, that may be more an aberration than it is the general rule, the simple characteristics that Gregor Mendel saw. But even Mendel himself realized this. He saw examples where his basic simple rules didn't really seem to apply. He realized this. He knew that his rules that he was formulating about genetics didn't necessarily apply to all crosses that he saw and all particular outcomes. But he didn't really have a way of studying this further. So his general basic rules are very applicable, and they help illuminate quite a bit in genetics. And without his knowledge and without his work, we would have been much further behind than we were. So he is considered the father of genetics, even though there's a lot that goes on in hereditary information and gene transfer that doesn't necessarily follow the simple rules. So we're going to take a look at some of those. The relationship between the genotype and the phenotype, especially, which is the genetics and what you actually see, is rarely as simple as what Mendel saw in the pea plants. Many heritable characters are not determined by only one gene with 2 alleles. So unfortunately, it gets more difficult because a lot of what you see, characteristics or traits, are not determined by a single gene and only 2 alleles. It can be far more complicated. Say, for example, hair color in humans is likely determined by a number of genes, not just one. However, the basic principles that Mendel came up with of segregation and independent assortment, they do apply even to these more complex patterns that we're going to take a look at. So let's start with some variation with a single gene. So in Mendel's peas, a single gene was either dominant or recessive. There were only 2 possibilities, 2 alleles, and that was basically it. When we were talking about a single gene, he didn't know it, but located on a single chromosome, and it was basically on or off, purple or white, green or yellow. That's nice. But in the real world, it's actually oftentimes not that way. In humans, for example, again with hair color, it's obviously not just black or blonde or brown or red. There's all sorts of different variations between as well. So let's take a look at that. Inheritance of characters by a single gene may deviate from the simple Mendelian patterns in the following situations. Say, for example, when alleles are not completely dominant or recessive. Sometimes there's not a complete dominance or recessive. When a gene has more than 2 alleles. So what if there is a third shoe in our pair, a third possibility? And then finally, when a gene produces multiple phenotypes, what if one gene produces different looks based on different conditions? That's what we're going to take a look at. The first one had to do with dominance. There can be, in many cases, in many genes, degrees of dominance. Complete dominance occurs in the Mendel's peas that he was looking at, where the phenotypes of the heterozygote and the dominant homozygote are exactly the same. So it didn't matter if the pea plant was homozygous dominant, it had purple flowers, or heterozygous, it still had purple flowers. So the dominant is completely dominant and the recessive is completely recessive. That is simple, but that only occurs some of the time in what we call complete dominance. There's another form called incomplete dominance, and the phenotype of the F1 hybrid is going to be somewhere in between. And that would be, if in Mendel's case that occurred, it would be a purple flower and a white flower, and you would get, say, a lavender colored, a light purple colored offspring. That would be incomplete dominance, where you have this blending. Remember that blending theory we first talked about? You might actually kind of see this in that particular case, or it would appear to be like things were blending, but we call that incomplete dominance. The last one is called codominance, and you can probably guess based on the word. Codominant means the 2 dominant alleles affect the phenotype in separate, distinguishable ways. In other words, codominant means you actually create a third possible phenotype. It's not a mix or a blend like an incomplete dominance. it could be a third completely different look or phenotype. Let's look at an example of incomplete dominance where you kind of see that blending effect. So this is in a type of flower called snapdragons, or a plant called snapdragons. There is a red and there is a white. If this, say, example, they have two variations of the allele, so they put a little R for the red, a little W for the white. When those two pure breeding flowers are combined, they create a hybrid. But the hybrid doesn't come out to be red, which is the dominant. It comes out to be pink. So this is incomplete dominance. We have kind of a blending of that. Now, what if you were to take two of the hybrids and breed those together? Well, you would get not the 3-to-1 ratio that we get in complete dominance. You would get a 1-to-1 ratio, which is what you see with this incomplete dominant characteristic. If red was completely dominant, we would expect to see these 3 red and 1 white. So this is what might cause people to have thought about this blending hypothesis, this blending idea of genetics, this generation, F1. But then when they put those together, you would definitely expect them all to be pink. And then it really gets crazy, because if you expected this to be blended and then 2 pinks to create just more pink, well, you'd get a red and 2 pinks and a white, and that would really kind of ruin your blending hypothesis. This idea of dominance and recessive is nice and clean if it's a complete dominance, but in reality it's not like the dominant allele sits on top of the recessive allele and hides it or covers it up. They don't really interact in that way. One allele doesn't directly affect the other like that. The alleles, remember, are simply slight variations in a gene's nucleotide sequence. And for any particular trait or character we're looking at, whether you might call it dominant or recessive, it kind of depends on how you look at it, or the level at which we examine the phenotype. So let me show you what I'm talking about. There is a disease in humans called Tay-Sachs disease. It is a fatal disease. It's a dysfunctional enzyme that causes an accumulation of lipids, fats in the brain, and it can lead to your death. So if you have Tay-Sachs disease, you have an enzyme that doesn't work inside your body, and that allows for this buildup that can eventually cause problems and lead to your death. The disease is only seen or manifests itself when the child is homozygous for the trait. In other words, the child has received the allele for this mutation from both parents. All right, so let's take a look at the organismal, that's hard to say, organismal level. So if we just look at the whole human itself, the allele for this disease is recessive because it doesn't show unless 2 copies of the allele are present. So that would seem to indicate that it is recessive because it only shows when you get 2 copies of the allele. If it were dominant, it would show up with only 1 copy, and it doesn't. So we would call that recessive at the organism or the organismal level. At the biochemical level, the phenotype, again, that would be the enzyme activity, that's the phenotype we're looking at, whether or not the enzyme works, is what we would call incomplete dominant, because if you are heterozygous for this trait, for the Tay-Sachs allele, you have half the functional enzymatic activity as a normal person. So if you're heterozygous for this trait, you produce about half of the enzyme that your body needs. So you're midway between producing all the enzyme like a normal healthy person and no enzyme like a person with full-blown Tay-Sachs. So at the biochemical level, you would be considered, this would be considered incomplete dominance, because you can be heterozygous and then you fall somewhere in between. So that would be like incomplete. At the molecular level, the alleles, you would call them codominant, because if you are heterozygous, at the molecular level, you produce an equal number of working and non-working enzymes. So they don't really affect each other. You have some cells that produce working enzymes and some cells that produce non-working enzymes, or the cells produce a 50-50 ratio of working and non-working. So we would call that codominant at the molecular level. So really it's the way you look at it, or the level at which you look at it, that can be considered dominant or recessive. It's not really clear cut, like when we were talking about Mendel's flower colors. Another important factor to remember that a lot of students kind of get confused about is just because we say an allele, a particular allele is dominant, that doesn't mean it's always the most frequent. So you can have cases where the dominant allele is present less often in a population. The dominant allele is not necessarily the most common. Now, oftentimes it is, but I don't want you to think that dominant allele also means most common allele. Not necessarily true. So a good example is what we call polydactyl. One baby out of about 400 in the United States, so that's not terribly uncommon. But obviously 399 don't have this. One baby out of 400 in the U.S. is born with extra fingers or toes, usually not fully formed, sometimes like just a sixth small finger or a sixth small toe on one foot. That's called polydactyl. Some of these cases are caused by the presence of a dominant allele. So in some cases, polydactyl is caused by a dominant allele that appears. So what that says is the recessive allele, having 5 fingers and toes, is far more common because 399 out of 400 people will have only the 5 fingers and toes, and that is caused by a recessive allele. So don't think that recessive means least common or dominant means most common. Sometimes that is the case, but that's not necessarily the case, and that's not exactly what it means. So we'll talk about how natural selection affects the relative frequency of certain alleles in a population, and we're going to talk about that more in chapter 23. Going back to Mendel's pea plants, he was looking at characters that had only 2 possibilities, 2 alleles, but most genes exist in populations in more than 2 allelic forms. So, for example, blood type. In humans, there's 4 phenotypes of the ABO blood groups in humans, and it's determined by 3 alleles for a particular enzyme that attaches a certain carbohydrate, A or B, to the red blood cell. So you can have a number of possibilities. So in the case of human blood type, you can have basically 3 alleles, and that actually is going to create 4 different phenotypes. But the enzyme is either going to be the IA, the IB, or the little I allele. So you have kind of like 2 versions of the dominant, and then you have the one recessive. So the IA allele adds this A carbohydrate to the red blood cell, the outside of the red blood cell. The enzyme encoded by the IB allele adds the B carbohydrate, and the enzyme encoded by the lowercase I adds neither one of them. So here are your 3 possible alleles. The IA adds the A carbohydrate, the B carbohydrate, and the lowercase I, the recessive, doesn't add either one of those carbohydrates. So how does that get you to 4 possible blood groups, genotypes and phenotypes? So look at all the various combinations. You can have 2 of the A's or 1 A and 1 none, and that would give you a A blood type. 2 of the B's or a B and a none gets you a B blood type. An A and a B is an AB blood type. Or maybe you have the none and the none, and that gives you the O blood type. So you have 3 different alleles. That's going to give you 4 possible combinations, and that gives us our basic blood types, A, B, AB, and O. And then, of course, you probably heard there's also negatives and positives, and that gets into another enzymatic action that we're going to not quite go into here. But this just shows you that a lot of times genes have more than 1 or more than 2 possible alleles. Here we have 3. Look at this diagram. It might also help explain why blood type O is oftentimes called the universal donor. And you'll notice it's because O has none of these carbohydrates sticking off of it. So pretty much anyone's body can accept this blood cell because it doesn't have any of these carbohydrates that might trigger a reaction. So if you had, for example, A blood and we transfused B blood into you, your body sees these carbohydrates on the outside, recognizes this blood cell as a foreign invader, and it causes a severe reaction. But the O blood type, seeing as it doesn't have any of the carbohydrates, it's not like the body can detect something that's not there. So the O is considered universal donor and can be pretty much given to all of the other potential blood types. So we talked about how in many cases there's more than just 2 alleles for a gene. Well, now we're going to get to something called pleiotropy. Let me make sure I'm pronouncing that correct. Pleiotropy, I believe. Or it may actually be pleiotropy. Pleiotropy or pleiotropy? My gosh, these science terms can be so hard. So most genes have what we call a multiple phenotypic effect, a property called pleiotropy or pleiotropy. It comes from the Greek word pleion, I believe, meaning more. And this is, for example, the gene that determines flower color in peas also affects the color of the coating of the outer surface of the seed, which can be gray or white. So in pleiotropy or pleiotropy, one gene actually has multiple effects and affects multiple phenotypes that we see. So another good example that we list up here is pleiotropic alleles are responsible for multiple systems of certain hereditary diseases, such as cystic fibrosis and sickle cell disease. So it's one allele, but it is causing multiple issues. It has multiple phenotypic effects. So one gene affects multiple phenotypic outcomes or things that we see. So that's pleiotropy or pleiotropy. I'm not sure exactly how to pronounce that, but I would take either one. So let's now extend some of those variations to 2 or more genes. So we were just looking at the variations that might affect one particular gene. So a gene that might have multiple alleles, or a gene that has basically multiple effects, multiple visual effects that you might see on the physical characteristics of the creature. Now let's kind of talk about some of the differences that Mendel didn't see or didn't study that involve more than one gene, 2 or more genes. And the first one we're going to look at is the fact that some traits, or actually many traits, are determined by 2 or more genes. So in humans and actually many living creatures, a lot of what you see as far as their physical characteristics weren't determined by a single gene with 2 alleles. Oftentimes it's multiple genes that can affect the phenotype of what you actually see. And a good example is in lots of the dogs' hair color, particularly like in Labrador retrievers. You have 3 major, I guess, varieties of Labrador. You have black Labradors, you have chocolate Labs, and then you have yellow Labs. And these colorations that you see are actually determined by more than one gene. This particular variation is called epistasis. Epistasis is where a gene at one locus, one location, actually affects the phenotypic expression of a gene at a different locus, a second locus. So in Labrador retrievers, this is the case for determining their hair color, whether it's black or yellow or kind of the brown chocolate in between. This is also the case in many other mammals in their coat color and fur color. Depends on 2 genes. One of the genes determines the pigment color, so that was either going to be black or brown. So capital B is dominant black and lowercase b is recessive brown. And the other gene with allele C for color, capital C for color, lowercase c for no color, determines whether the pigment is going to be deposited into the hair. So you have 2 genes going on. One of them determines color, and one of them determines whether or not that color is going to actually be deposited into the hair. So take a look at a cross that takes into consideration these 2 factors. So if you see, and these are all the different possibilities based on the fact of Mendel's various genetic rules and all of the variety that we can have. If you see anything that has a capital B in it at all, whether it's 2 capital B's or a capital B and a lowercase b, that's going to code for black fur color, because black is dominant. If you see 2 lowercase b's, that's going to code for the brown or chocolate fur color. Now, in the E, if you see at least 1 capital E, it's going to deposit whatever the B is. So like in this case, you have homozygous dominant in everything. So it's going to deposit whatever the color is that's black. In this case, how about this one right down here? Well, it has a capital E, so that is the dominant and deposit the color. So it's going to deposit whatever the color is, lowercase b. But what if we get homozygous recessive for the deposition gene, the E in this case? Or is that a C or an E? It is an E. So if we have 2 lowercase e's, we're not going to deposit the color. We're not going to deposit the pigment into the fur. So in reality, if we have 2 lowercase e's, we're always going to be blonde or yellow in the case of Labs. And it doesn't even really matter what the coloration letter is. So that second letter determines whether or not the color is going to be applied to the hair, and the first letters determine what color is going to be applied. So that comes out with 3 different possible traits. And this is kind of an odd ratio of a 9-3-4. Because if there is Homozygous recessive for that second allele, it doesn't really matter what the first allele is, because that second allele kind of turns off the pigmentation of the fur no matter what. So all of these are going to be blonde. It really doesn't matter if they have the brown coloration or the black coloration. So that's called epistasis, where one gene at one location actually affects a different gene at a different location. And in labs, these are our choices. If the dog has a capital B and a capital E, that color will be expressed and the color will be black. The chocolate lab is going to have the lowercase b's for brown and at least one capital E, and that means the color is going to be expressed brown. And the yellow labs have the recessive two small e's, which means no coloration for them. It doesn't really matter what their color, big B or little b, was going to be because it's not going to get expressed anyway. So these are the three possibilities that you get in labs with this idea of epistasis, where one gene affects another. Okay, I see my mistake here too. I was putting C, and in all of the photos there, it's actually showing E. So I'm going to change this to an E and a lowercase e. Why they would use E for color, I have no idea, but that's what they did in the pictures. So I'm going to change my notes to match. So the allele for putting down the pigment or not, depositing the pigment or not, was capital E and lowercase e. That way we don't cause confusion. So that's epistasis. Let's take a look at another possibility with multiple genes. The other possibility with multiple genes that didn't really fit what Mendel was seeing or what he was studying is called polygenic inheritance. And polygenic inheritance basically means that you're going to have an additive effect of multiple genes, and you're going to end up with a wide spectrum of phenotypic possibilities. Instead of just the two possibilities that Mendel studied, purple or white flowers, smooth or wrinkled peas, you have this huge kind of rainbow of possibilities. And skin color, human skin color, is a great example of this. Quantitative characters are those that vary in the population along a continuum. So skin color is a quantitative character, and it's determined by a number of interactions with different genes. And this indicates polygenic, poly meaning more than two, numerous, polygenic inheritance, an additive effect of two or more genes on a single phenotype. So if you take a look at all these people down below, all these, I believe these are all young ladies from a particular class, you can see that human skin color has a huge range, a huge variety from very light to quite dark, and pretty much everything in between. So unlike the pea plants that just had, say, two particular possibilities of a character, human skin color is polygenic, where you have quite a bit of variety because there's multiple genes and they have what we call an additive effect. If we were to look at, say, a Punnett square that tried to explain all of this, you have all these different possibilities, and then you have all these different various combinations. And notice that each different combination sort of gives you a slight different variation, ranging from the very dark skin to the very light skin. So these are a number of different phenotypes. They show 1, 2, 3, 4, 5, 6, like 7 possible different phenotypes. And the phenotype you have is dependent upon the genetics that you inherit, numerous genes affecting this phenotype. And of course, this doesn't even talk about the environmental factors that can also change skin color. So we're not talking about whether or not you lay out at the beach and get a tan, or you during the winter you stay inside and your skin lightens. We're talking about the genetics that you receive. Generally, when something is polygenic, like a trait like this that is polygenic, it also has a wider possibility of change from the environment. So certain traits like hair color and skin color, they can be affected quite a bit by the environment. And traits that are polygenic, that have a wide variety to begin with, tend to be affected by the environment more than certain other traits. So for example, your ear lobes, whether they're free or attached, that is not really affected by the environment. It either is yes or no, free or attached, A or B. That's a Mendelian trait, which doesn't have a lot to do or isn't affected quite a bit by the environment. So talking about polygenic inheritance can kind of lead us into this whole conversation of how does the environment affect genetics, or how does the environment affect phenotype? because it doesn't really affect the genetics, but it does affect what you see. It can affect the expression of those genetics. So here's another departure from what Mendel studied, where the phenotype can somewhat depend on the environment as much or as almost as much as the actual genotype. We have something called the norm of reaction. The norm of reaction is the range that a genotype will provide. So you could have a particular genotype for skin color, and that will give you a range. So if you're like me, you tend to get darker in the summer when you're out in the sun more, and you tend to get lighter in the winter when you are inside or you're exposed to less sun. But there is a certain range. I'm only going to get so light even in the winter, and I'm only going to get so dark even in the summer. That's the normal reaction for that particular trait for me. That is determined by my genotype. But where am I within that range? That is going to be determined by my environment. Another good example in gardening is hydrangeas. If your mom or dad are gardeners, they might like the type of flower called a hydrangea. And that particular flower can be found in multiple colorations, blue, sort of violet, to pink. And that actually depends on the soil acidity. So if the soil is somewhat basic, meaning the pH is above 7, you're going to get pink sort of colored flowers. If the soil is slightly acidic, meaning below 7, pH 7, you're going to get blue colored flowers. So you can actually affect the color of your flowers based on what you add to the soil or the soil type where the plants are growing. Like I said earlier, the normal reaction, or the range that you might get, is generally broadest for traits or characters that are polygenic, so those that are controlled by multiple alleles. So again, like your earlobes, whether they're free or attached, that's only controlled by a single allele, and it's not going to really be affected by the environment. But skin color, which is controlled by multiple alleles, has a broader possible norm of reaction, possible range, and is more greatly affected by the environment. These characters that are affected both by the environment and genetics are called multifactorial. Multifactorial meaning there's multiple things that can cause them to look differently. You have the genetics, which are kind of like your starter blocks that you began with, but then you have the environment, which can also cause changes and influence the phenotype. And that gets you to that whole debate of genetics versus environment, or nature versus nurture. There are certain characters or traits about creatures where genetics play the primary role, and then there are certain traits or characters about creatures where perhaps environment plays just as important a role, or perhaps even more important. So let's try to kind of wrap up what we learned from Mendel with what we know now, because we know so much more than Gregor Mendel did, but still some of his basic rules are very applicable, and they help us understand genetics even with the greater amount of knowledge that we have today. So we want to make sure we understand that an organism's phenotype includes all aspects of its physical appearance, anatomy, physiology, behavior. When I say phenotype, I don't just mean the way it looks, but there's a number of other characteristics or traits about that creature. That could be things you don't see on the inside, or it could be the way that the creature behaves. All of those could be considered phenotypes, or the overall phenotype of the creature. The phenotype of a creature will reflect its overall genotype and its environment. So the way a creature behaves, the way it works inside, the way it looks, that is a product of a number of factors, including its genetics, but also including environmental factors. And Mendel's theories of inheritance can still be applied to what we know today, and they are also applicable to all living creatures that have a sexual life cycle. So it's amazing that these genetic laws or rules that Mendel kind of developed, they apply to humans, but they apply to pea plants and whales and oak trees and aardvarks and guinea pigs. They apply to any living creature that has a sexual life cycle. And that is pretty amazing, that these characteristics or these rules that he determined and discovered are applicable to almost all life, at least all life that reproduces in a sexual way. And that's pretty amazing. Again, that goes back to this overall idea of evolution and common ancestry, and how these genetic rules are applicable to something that seems so far apart, like an oak tree and a human. It still works pretty much the same way. And again, the reason I love Mendel and his story is that's pretty good to develop these ideas that have withstood the test of time and even have been strengthened by what we know today. For a monk, an amateur scientist in a garden full of peas, that's pretty amazing, the information, the breakthroughs that he discovered. That's why he's called the father of genetics. He's a great example of where curiosity and the scientific method can take you. You don't have to be a professional scientist to add to our knowledge of the universe, and that, I think, is somewhat inspirational. So Mendel did his work not because it was his job or because he was trying to become rich and famous. It was just a passion for science that he had and this deep inherent desire to know more about the universe he lived in. And that, to me, is very cool. We're going to take a break right there. When we come back, we're going to finish up this chapter with Concept 14.4, which is all about human traits that follow Mendelian inheritance and some of the human-related genetics.